And yeah, so welcome everybody. Hope you have had a lovely week. And now today I'm going to talk about statistics, reproducibility, and also a little bit of software engineering that is very useful when you do projects like this. So let's see, get things running. So now I'm in the right place. So the literature for today is well, a little bit of the morphometry that we have always seen. John Ross. Hypothesis testing is something that is coming in a lot now, or is a central topic. I'm not going to go very much on the depth of it, but more giving you the directions. Also, graphics is something very important, and I'm going to spend quite some slides on how and why and the principles of visualizing your data. Then there are a couple of videos, podcasts that you can listen to. The first one is more typical lecture. The second one is more of the entertaining nature. So a guy who is bringing up all the fun conclusions people can do with statistics, and well, it's an entertaining 20 minutes to listen to. And then we have also a couple of slides, nice reading, some serious, some less serious, but it's anyway good reading to get up the critical thinking in what you're doing with your data. And then the data set that we are going to play with a bit is the IRIS data set, which I'm coming back to in a few slides. And well, let's go on. Also some papers and sites about unit testing, how to work with databases, also how to work with visualization. So there is a lot of material to read if you want to go into depth of different things. Until now in the class, we have been talking about pretty many things now. We are going towards the end. So starting out with defining images and how to enhance them, reduce the noise, enhance features that we want to look at. Also looking into the histograms, understanding grade level distributions, going towards automatic segmentation methods and labeling, and using that information, we now are able to do some shape analysis and also doing more complicated shape analysis, bringing us towards the dynamic experiments, which is going actually more towards the big thinking, and that we have a lot of data. Dynamic experiments are time series where you acquire information from different timestamps. So now we are going towards what are we going to do with all this information. So with the component labeling and shape analysis, we actually reduce the terabytes into hopefully some gigabytes, or possibly even mega or kilobytes. But from that, we now have to extract some conclusions. And that is about today's lecture. The big imaging, well, it's not only just about big, it's also about how to handle it. And the important thing is we have different things, how the data, first of all, how the data is getting big. And we have volume, so the amount of data itself, acquisition speed, and also variety. Very often you want different individuals to be investigated using the same repeated experiment, and that also produces the variety. So we need to have something that is scalable and hopefully fast, because if you're starting working on terabytes of data, you don't want to wait 2 years, because that would probably be too late for finalizing your thesis. So you need something that is also well worked through, that it can work fast. So to do this, we need to work in a very disciplined manner. First of all, we need clear steps and that are isolated, so it's not too nested in each other, because then it's much harder to regenerate what you did before. So by having these fixed steps, you can also go back only one step or two steps and starting from an intermediate result, in the case you save the data, of course. For sure, there shouldn't be any magic in your analysis. So that you need to do 3 loops with a mouse and double click in order to get your results. And very important is to document your analysis, what you did. And for that, our Python notebooks are very useful because you can actually read or actually write in description of the steps you have performed, as you have seen also in my lecture notes and also in the exercises that I've written, sometimes pretty long text blocks. You can even use LaTeX to get the equations, and with that you have a very nice workflow and that you can work through. That is good for the stepwise documentation. If you have, on the other hand, say core functionality, then that should be put into separate function files, which can also be very good documented, at least what the purpose is. So the important thing is that you should have prepared your analysis in a way that it should be, for you at least, as easy to process 100 samples as a single sample. Of course, the computer will probably sweat a lot with 100 samples, but that's not your problem. You only have to wait, and then you can do something else. So it's important that you can actually scale up your analysis. And with that, we should also have the possibility to be flexible enough to work well with different parameter sets or different types of cells. Of course, the analysis is usually tailored for a specific type of analysis, so you can't say that if you can use the same process in analyzing cells as you would do with elephants. Probably the questions are completely different, so you can't reuse it in that sense, mostly. Yeah. So in scientific studies, we try to get single numbers describing how something behaves. So it could be, and then when you do this, you need to have something that describes what you want to do in a kind of clear manner. So there shouldn't be any confusion. And also the next step is making it meaningful, and that is what we're going to talk about today. So talking about making conclusions out of it. Also, in this sense, we want to have a possibility to compare numbers of different samples, different sample groups, etc., and compare if they differ depending on the treatment of the data. And that could be also be working on the processing itself, so seeing what is the processing doing to and actually also going back even to the acquisition and the whole experiment design. How does the experiment design affect the outcome of your experiment? So that is also one thing that we're going to look into, is scanning over parameters in the analysis sequence. And of course, in the end, you have to present your information in some way. And that is where the visualization come in, and that is the last part of today's lecture. Now I missed also number 5, how we can ensure that our techniques are doing what they're supposed to do. That's also within this parameter sweep evaluation. So that is essentially bringing it into bullet points more compressed manner. So let's go on. The first thing is we want to look at our cell image, and that one we have been looking at pretty many times already within this class. So first of all, how we work with the images, how to extract the information. And now we have some data and we want to do something with it. And the question is, in an experiment, what kind of experiment you have. So you, on one side, you want to do correlation. That's a statistical relationship between different samples or groups or something. It's very easy to show. It's a very simple calculation to do in the computer. A more difficult one is the causation, is what happens if I introduce a mechanism? Is there some connection between group A and B caused by a mechanism that you have applied to them? And that can be, depending on how you design your experiment, can be very difficult to show, but also in some cases even impossible. So you have to break down your questions in your project in a way that you can show at least subsets to make the conclusion that the causation actually is present in this experiment. For that, we have two different types of experiments. So we have the observational experiment, so where you just look at what's happening. And then you have the control experiment, where you prepare samples in a certain way. In more detail, this is that we explore large data sets, looking for trends. We just look in the world and see how wonderful it is or something. And then we see if there is a trend in why it's wonderful. The population is mostly random, but it can also be selected, but then you're going towards the control experiment already. It's not always hypothesis driven, and it's difficult to see any causation in that. Possibly, again, if you ask your questions in the right way, you can see it, but then we are possibly going into dangerous conclusions. So there are some examples. We examined 100 people. It appears that the people with the blue eyes, they are on the average 10 centimeters taller. Probably in that case, Northern Europeans, or originating from the Northern Europeans, which tend to be taller than other people. Then there is another case where we have done a lot of baking. Then we see if we bake 100 cakes, we see that there is a 0.9 correlation between how long we bake the cakes and how big the bubbles are. In the control experiments, which is actually what very many scientific studies are doing, then we have the ability to control the specific details about the groups that we are investigating. So we are, for example, preparing samples in a specific way. We may add different process parameters, like having different temperatures. And with that, we can really see the causation between what happens if we add this component to the data, to the experiment. Example of that, we have, for example, 50 mice with gene XYZ off and 50 mice with it activated, and we see that the foot size increased by 10%. That's an interesting gene, by the way. Then we can also talk about looking at the temperature and the number of pores in a metal melt, and that they increase by 10%. So that's always that we modify something or separate the groups into specific characteristics and control what we are doing with it. So looking at a simple model, we have a magic or weighted coin. Magic for those who believe in magic, but probably more likely someone tampered with it and you get it to do something. So it's about tossing a coin and see how many times you get a head or tail. So there are different kinds of questions you can ask, and in order to prove if it's fair or not fair. So let's start flipping the coin 10 times, and then you let some other person flip it 10 times, and you ask if is it the same than if the same person would flip 20 times. You can also say design the experiment in a way that you have you toss the coin 10 times and then just multiply it, and then you say, okay, I have 100 times, 100 flips. Not really reliable statistical analysis. You can also do an expanded experiment where you buy 10 coins and you want to know which ones are fair and how you should design the experiment with that. Now, oops, there are some formatting problems here. So we have the coin. Each coin represents a stochastic variable, and each flip that we do is an observation of that stochastic variable. So we have the variable xi, and the observation is xi. The act of flipping the coin is f. So putting it into an equation, we would say that xi is the same as f applied to the stochastic variable x. In a fair coin, you would hope or expect the expectation of the variable to be a half. That means you should expect 50% heads and 50% tails. Also, if we have an unbiased flipper, that means that each flip is independent of the other. Then we can also say the probability of the multiplied, or the collected probability is the same as the probabilities multiplied by each other. Finally, also, we should have the expected value of all the flips to be the same as the expected value of the sample itself. Then we have a fair coin. So what we have is a little problem now when we go from the simple model to the reality. So coin flips, then each flip give us a small piece of information about the coin. That's good, but also the flipper, so the modification we do. So there is something happening with it. And to put it in more general terms, we have random variations. If we have random variations in the coin and the flipper, they should cancel out in the long run. But if we have systematic variations, they will accumulate. So for example, if the coin has something tampered with it, or the flipper is very good at producing heads, then we will see that in a statistical analysis. And going to the real experiment, the experiment itself, or the measurement tells us about something about the sample, of course, also about our instrument, but also about the analysis we have performed on the data. So more experiments provide more information, so we can start getting separating random and variations from systematic variations. So again, the random variations, they are in sample, instrument, and analysis. They should cancel out. And also the analysis has usually very little stochastic variation. It's pretty deterministic analysis that we are doing. There are cases when the randomness is included in the analysis, but that is extreme cases. Yeah, well, if you have the big some big data and want to do the stochastic gradient descent, then you are actually selecting some parts of the data in order to come faster to the solution. So then you would get some stochastic variation in the results. The systematic variations, they will accumulate. So for example, if you would set the threshold in the wrong ways, you would get maybe larger volumes of things or different counts of items. So that would be more the things that where the analysis would contribute. Let's look at some real data. Now we take a look at data of the flower iris. And we have a number of measurements in the data set. We have measurements from different plants and also different species. So that's the variations we have in the data. So let's load some data. We can see that we have different species here, different individuals. They are all having their different ID numbers. You can see that there are different length and width measured in this data. And let's bring up the whole statistics and see what we can get. Apparently this one wasn't executed. Oh, sorry, I have to go back a little bit. Yeah, there it comes. And there we get it. So now we can see that there are different, in this pair plot, we can see that there are different groups clearly. We see the species as different colors, and you can see how the histogram of each information piece looks like. And you can also see that in some combinations, you have better separation than others. So that is what we are going to look into now. And then we'll see, we can, one thing we can check is the intra-class correlation coefficient. And that is a metric that we can use to check how similar objects within the group are, but also if there is a similarity between groups. And that is something I need to execute. So let's look at the sepal data. Sepals are the leaves around the flower that actually in the beginning, when the flower is not yet ripe and opened, it's covering the flower. And in that one, we can see that the width of the sepals is not very much varying between the different species. So that would be a low group similarity. Looking at the petals instead, we can see that there is a high group similarity. So within the group, they are very clustered. And between the different species, there is a good separation. The petals, by the way, are the beautiful leaves of the flower. So that gives us two extremes in the data. And now I've only qualitatively talked about different similarities between the groups, but it can also be put into a metric. So the intercorrelation coefficient is defined as the estimate of variance among the groups or classes, and also related to the variance within the groups or classes. And you can see here that it's kind of a flipping of standard deviations. So in this case, we have standard deviation of the mean values in the group. And the other one is standard deviation, the average of the standard deviations in each group. And that is put into this metric here. And if we have a 1, it means that the variances between the classes is very high, so it's well separated. And a 0 between 0% variance between the classes is that they are almost the same. And let's look at what the group similarity is telling us. In the case of the sepals, we have a relatively low value of the ICC, while in the case of the fetals, we have a very high group similarity. So this is a way to separate if the variable is telling us, is able to separate between different classes. So now we are going to do some tests on this. Let's see how it's possible to compare between different groups. And first we have to, in the statistical analysis, we have to make a list of different assumptions. From those assumptions, we have to establish a null hypothesis. Usually what we are asking for is, are both groups the same, or is there a difference between them? And then we need to calculate the probability that the observation, given the truth of the null hypothesis, is fulfilled. And with that, we need knowledge about the probability distribution. And there is a little bit of a problem here because it's not always you know the distribution, and that can be very complicated to get it. In many cases, the quick fallback solution is assuming we have a Gaussian distribution, which is more or less true if you look at the law of the large numbers. At some point, any distribution will turn into a Gaussian, or can be analyzed as a Gaussian distribution, but you need a lot of data for that. Look at the coin example again. We have this coin from the magic shop, and now let's put up our assumptions. We have we flip the coin fairly accurately and independently. The coin is also invariant and always has the same expected value. So our null hypothesis is that the coin is unbiased. That means the expectation value is 0.5. Now we can calculate the likelihood of a given observation to be within this distribution. And let's look at the Gaussian curve. We can see that we have the curve where we have the most likely observation at the peak of the distribution. And then we have tail observations where it's very unlikely. If we have a probability out here, it's very unlikely that the coin is actually a fair coin. And the p-value is a threshold that tells us, or we have to compare to the p-value, so the probability of the coin being within or not is our threshold. So the question is, what is now good enough for our analysis? Very often we are talking about 95% within is a good assumption. Also, another thing is, if we want to do hypothesis testing, we can also use the student t distribution, which is developed sorry, which is developed for the case when you have very few observations. So the student t distribution is slightly skewed for low numbers, but it moves towards the Gaussian when you have larger numbers, and it's very much used for analysis with few samples. And it assumes that we have a Gaussian distribution, and it also assumes that we have unbiased observations. So there's a couple of requirements to use this, but it's very commonly used within in the statistical analysis of hypothesis testing. So let's come back to the magic coin and see if we can do something with it. Of course, we would like to publish something, so let's make the test. We had p-value less than 5% is good enough. So that means if we toss 5 times and 5 heads, we are able to tell that this is clearly a bad coin or a magic coin. So let's look what we are doing. We have the analysis where we look at the probability of tossing 5 times in a row and getting also 5 heads. And as you can see in this table here, if we flip 1 time, the probability of getting a head is 50%. If we do 4 flips, then we have 6.4% probability. And if we have 5 in a row from a single experiment, then it's 3%. So with that, we are actually beyond the limit of saying, or we're actually beyond the threshold that says, okay, now we can reject the null hypothesis. And let's do a different experiment. Now we have many friends, and we let each friend make 5 tosses, and let's see what happens. Will we get 5 heads from someone? And if we only have 1 friend, well, the probability is still 3.1%. But if you let 10 friends toss the coin, then all of a sudden the probability is increasing. And when you have 80 friends that do the same experiment, the probability that someone gets a coin is 92%, gets 5 heads in a row is 92.1%. So in principle, if we have sufficiently many people to do something, it will happen. And that's also like the story about if you put a monkey on a typewriter and let him type randomly, at some point he will type Shakespeare's collected works in 1 row. Not very likely, but if you let him do it infinitely, at some point he would probably do it. So now the problem is we can't just let more and more people do things and say, yay, now we got it. So the p-value is essentially only valid or meaningful when we do what when the experiment matches what we did. So we didn't say that the chance of getting 5 head ever was less than 5%. And what we said is exactly 5 observations and all of them are heads. That is less than 5%. And that is also our rejection criteria for saying that it's a fair coin, or that the likelihood that a fair coin produced this result is less than 5%. So we need to correct the whole principle based on this knowledge. So what people are usually doing is to scale. There is also something weird happening. So to scale the p-value with the number of experiments. So that's bad, kind of. So we can quantify a lot of interesting metrics, and but we have to do the analysis in a very well-designed way, and also we have to look at the large numbers and also do the correct analysis on them. And let's see how increasing numbers affect a number of variables affect the rejection rate, or actually, let's see here now. Let's create some data. We have just a bunch of random variables. In this case, it's a uniformly distributed data. And let's see what happens with that data. So it seems like we only have 1 that is significant among 5 variables here. But if we increase the number of variables, that would be the number of friends that you have that start tossing coins. Then you can see that, oh, okay, in this case, we have out of 20, all of a sudden we have 2. And that could be significant, significantly outside of the null hypothesis. And let's expand this a little bit. You can see that if you increase the number of variables in the study, you can see that there is a steady increase in the number of significant values found. And so this indicates we need to do some kind of scaling in the data, in our analysis. This is another distribution. You can see with box plot instead. You can see the variation, and then it's maybe easy also to see the trend line. And a typical way of doing this bias correction is to take the p-value and divide it by the number of tests and see what comes out. And if we go back again, you can see that we have these 5 heads. Now let's see. So there is this trend. I would say it's a pretty low slope in this case, but it's probably also because of the uniform distribution. But anyway, let's go and do this scaling. And you can see here now that we have the number of significant values found is increasing steadily. And now we do a scaling with the number of p-values. And you can see that then with that, you can have more or less the same significance level, also with larger number of experiments. Now we did something on a single, where all variables are the same. But now we have a case when we shift the data of one single variable by 1 standard deviation and see what happens with the p-values then and the significance in that case. So let's see if I can make some more space here. Yep. So with this, you can see here that if we have one variable and doing this multiple correction, well, divided by 1 doesn't make a difference. So the p-values are usually in the low range here. But if we have one deviating variable out of 18, you can see that something is happening out here. And that goes on for even more. When we have more variables, the effect of the deviating variable is getting stronger and stronger. And at the end, it's pretty strong out here at 87 variables. So a single variable that deviates and is biased, and it actually comes out pretty clearly. Let's see. No. So the likelihood of detecting a really different variable decreases with the number of variables, according to this plot here in this particular experiment. Now, let's change a little bit. We have now just done the normal prediction using the Gaussian curve and hypothesis testing, but there are so many different ways of doing this. And in scikit-learn package in Python, they have pretty big forest of different categories, even of data. And this decision tree here is supposed to help you to select the right method. So starting here, it starts saying if you have less than 50 samples, please get more data. Okay. And then you have to select if you want to predict a category, yes or no, or you want to predict a quantity. And in the end, hopefully you will end up in one of these green leaves. But there are some cases that scikit-learn is not supporting, and then they tell you tough luck. But for many of our use cases, I think we are well served with scikit-learn. But there are also other packages that could be used, or in the worst case, you have to spend some time and develop the kind of analysis that you need. In particular, if you have very many samples, then it could be that you need to do something special of your own, in particular for the clustering and structure prediction. So when we do our analysis, we have different inputs. We can have an image or multiple images. We can have a single point. We can have a feature vector, even a multidimensional tensor. And the output you want may be a value, some classification, a group, though that would be that we want to do some clustering, but it can also be a vector matrix with fewer degrees than what you put in the data. So it's a kind of dimensionality reduction that you're doing. What can happen is that you produce overfitting. So if you have a classifier, in this case, we do a magic classifier that tells that dog is an animal, Bob is a person, fish is an animal, and we apply that to the same data. You should hopefully produce the same result. So that dog is an animal, Bob is a person, and so on. So we do perfectly with if you use the training data directly. Now, if we try on new data, we could say that, well, octopus is an animal. We didn't train that. So it's fantastic. Wow. Working fine. Dan is a person. Even more fantastic. So it's this kind of exception. So what's happening here is the training looks like some kind of indexing in the data set. So it's overfitting. It's giving you too detailed answers, or really sometimes even the wrong answers. And it shouldn't work without outside of its own group. You can get extreme mispredictions if you go outside of what the classifier was trained for. That's the problem with the overfitting. So we need to do some validation. And I think, oh, it's 10 o'clock. So I think let's leave it here, take a break. I think I need to drink a little bit. Should we say 5 minutes, or you want more? Well, let's do 5 minutes break, and then see you again.